Hello. To go through the assignments, I will start from the very easy question first. We will go with the first molecules, which is quite simple in a sense. Okay, this is the first molecules of matrixyl. Matrixyl is an oligopeptides. Okay, so what I ask you to do is to find that how many amino acids included in these molecules and where is the peptide bonds, and also to identify the terminal nitrogen of amino group and terminal carboxylic acid group, N and C terminus. Okay, to make it easy, you have to know the structure of amino acid. Normally, amino acid contains carbon in the center, linked together with carboxylic group and amino acid group, and then we have R group. So after that, this amino acid connect with other molecules. Okay, but anyway, the character of this, you have to find this one, which is called alpha carbon. Okay. I will label all the alpha carbon in the center of amino acid. Then you can count the number of amino acids very easy. So the alpha carbon is the carbon in between amino group and the carbonyl group. N and carbonyl. So this is the alpha carbon. And this is belong to the first amino acid. As you can see, amino acid and the side chain. This is lysine. Next alpha carbon is here. As you can see, we have carbon in the middle between nitrogen and carbonyl. Carbon in the middle of nitrogen and carbonyl. Carbon in the middle of hydrogen and carbonyl. And carbon in the middle of hydrogen and carbonyl. So, as you can see, we have 1, 2, 3, 4, 5 amino acids. So this matrixyl composed of 5 amino acids. But 2 of these amino acids are the same. This one and this one are the same. So we have 3 types of amino acids, but in 5 molecules. Okay. Now, we have to find, as I said, the peptide bond. Peptide bond is the amide bond linked between 2 amino acids together. So as you can see, we have first amino acid over here, second amino acid over here. In between, we have amide bonds between 2 amino acids. So this amide bond is called peptide bonds. So we cross out the peptide bonds over here. So this is the first peptide bond. Now, second peptide bond, you can find it in between 2 dots, right? Because this is alpha carbon, this is alpha carbon of the second and third amino acid. So in between, we have this one, which is the amide bond. So the amide bond that linked between 2 alpha carbon together, this is the peptide bond. Next, 2 of dots, you have peptide bond in between. 2 of the dots, you have peptide bond in between. So, in summary, you have 1, 2, 3, 4 peptide bonds and 5 amino acids. So, as you can see, amino acids, 5 of the amino acids arrange themselves together in the linear shape, like this. So you have 2 ends, right? Left-hand ends and right-hand ends. Now you have to find whether left-hand ends is N terminus or C terminus, things like that. So if you look at this, alpha carbon contains amino group and carboxylic group. The carboxylic group is now condensed and connect to other amino acid. So N here is the terminal one. So this is terminal, N terminal or N terminus. But the N terminus over here is not appeared as free amino group. Because this is modified at the end with the fatty acid. Carboxylic acid with a long chain of hydrocarbon here is called fatty acid. You will learn about it in the lipids in the next topic. Anyway, this is fatty acid. So it's modified with our amino group. So N terminus is now modified. But for C terminus, as you can see, as I said, we have 5 dots, right? We have first dots and the last dot here. This last dot has nitrogen amino group and the carboxylic group. So N connect into the chain. So you have final carboxylic as the terminus. So this is the C terminus carboxylic group, but it's free from modification. So you have free carboxylic ending. And the amino ending is not free, it is captured by the fatty acid. So it's modified N-terminus. So this is the structure of the matrixyl. Now we're going to look at the next one. So can I just clear the structure here first? Now we're going to the next example, which is somewhat a little bit more difficult. Now again, if you have kind of struggled with that first one, you may have to take your times working on this one again. So you may pause the video now, okay, and then work with this one. But if you're ready, then continue the video. Okay, so first of all, in the same way that we already did with the first molecules, these TRF factors, you have to find the alpha carbon first. The alpha carbon is the carbon between N and carbonyl. This N is carbonyl. The carbonyl over here, and there's nothing in between, no? Carbonyl and there's nothing. So this carbonyl is nothing. N here, carbonyl and carbonyl over here. So in between you have a dot of alpha carbon. And then you have N here and carbonyl here. This is the dot in between N and amino group. So you have just only 3 dots, 3 amino acids, okay? And nothing in the same, right? 3 different kinds of amino acid. This one is called histidine. You have R group, which is obviously shown here. This 5-membered ring and this 5-membered ring are not the same. Because this one you have carbonyl group, but this one you don't have the carbonyl group in the ring. So we have 3 different kinds of amino acids. Now you have to find the peptide bond. As I say, the peptide bond is the bond that actually is the amide that linked between 2 amino acids. This is the first amino acid, this is the second amino acid. So in between, you have the peptide bond over here. Next, this amino acid and that amino acid, you have peptide bond here that linked together between 2 dots. So this is the peptide bond. That's it. So you have 3 amino acids, but you have only 2 peptide bonds, okay, to link them together. Now, the most difficult part of this question is how can you find N-terminus and C-terminus? As you know, we have just 2 dots at the ends, right? Left and right hand ends. This is the central dot. It's nothing. Don't have to look at it. So this one, you have N and carbonyl. But the carbonyl here linked with peptide bond. So N here have to be the final N, the N-terminus. So this is the N-terminus, but it is already modified with something. You don't have to care about it. But this is N-terminus. Now, for the right-hand end, you have alpha carbon. N of the alpha carbon linked with the peptide bond. So C of the alpha carbon here is the terminal carbonyl. So the terminal carbonyl is normally appeared as carboxylic acid. But as you can see from here, that it is not carboxylic acid at all. It is modified already with the ammonia. So making the simple amide at the end. So you have modified C-terminus and modified N-terminus. So this is N-terminus, this is C-terminus. Okay. So the way they modified and the structure of this may confuse you a bit, but yeah, if you just go from the very basic principle, finding the alpha carbon first, and then go to the peptide bonds. So it's quite simple actually. So everything is the same for every molecules of proteins. Now we're going to look at this one. A huge one actually. This is bovine oxytocin, as I said. So you have to find the alpha carbon first. Amino acid, amino group, and carbonyl. So this is the first alpha carbon. Amino group, carbonyl group, alpha carbon is in between. Amino group, carbonyl group, alpha carbon here, alpha carbon here, alpha carbon here, alpha carbon here. And then amino and carbonyl, alpha carbon. Amino and carbonyl, alpha carbon. And normally you forget about this. So So I've seen many students forget that this is actually an alpha carbon as well. Amino and carbonyl. This is alpha carbon, but the R group is just hydrogen atom, right? It's not like other amino acids. You have several fancy type of side chain, but the side chain of this one is just hydrogen atom, but that's fine. Now we have how many? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 amino acids in the system. So we call this as nonapeptide, right? Because it's the nonomer of the peptide compound. Now, between two dots, this is the peptide bond. Between these two dots is nothing, right? Between these two dots is peptide bond, right? Amide between two dots is called peptide. Amide between two dots is peptide. Amide between two dots. Again, amide between two dots. Two dots, the amide bond. Two dots, the amide bond. That's it. We have 1, 2, 3, 4, 5, 6, 7, 8 amide bonds. And the special thing of this kind of protein is linked into cyclic structure. The link between this unit and this unit is the disulfide bond that I already mentioned it earlier in the lecture. So this is a disulfide bond that linked between the cysteine. This is the side chain of first amino acid, and this is the side chain of this amino acid. Cysteine and cysteine condensed together by disulfide bond bridging. Okay, so as you know, again, we have two ends, right? This dot and this dot is the ending. Okay, where is the final end? Where is the end terminus? You may see both of the ends contains NH2. So go back to the principle, go back to the other compounds that I already showed you. So this is the last dot, right? The first dot or the last dot we don't really know, so we have to find. This dot, you have N and carbonyl. Carbonyl have, this carbonyl links with peptide bond already. So this one shall be the N terminus. Okay. And for that one, N is linked down here with the peptide bond. So carbonyl is the C terminus. But the C terminus here is not simple C terminus, it's not carboxylic acid, because it is now modified by the amine, by the ammonia. And you end up with that simple amide bond, right? So that's it. Quite simple. Okay, so we can just discuss about the structure of several oligopeptides. Now, I want to show you the proteins in action for the last question that I just gave you in the last lecture, in the previous clips. So, look at this one. Okay, as I already mentioned earlier, that this is the structure of ADP and a phosphate that is going to be linked together by the enzyme called ATP synthase. So ATP synthase is a huge enzyme. Okay, it's a huge enzyme, and there is a pocket. Okay, that the DNA, that the ADP is inserted down here in the pocket, and then it's now ready to link or condense with another phosphate group of the phosphoric acid. So after condensation here, you will have the ATP as a product, adenosine triphosphate. Okay, so actually the center of the reaction region is here, because you have to condense two of this unit together. So here, around here, have to be your active site, or the activity, or the catalytic site of your enzyme. But as you can see here, if the enzyme gonna catalyze one particular reaction, you have to, the duty of the enzyme is normally fix two of the substrate together, make it coming close together into the right direction and collide them together. So as you can see, the right hand part, the whole right hand part over here is used to capture the whole molecules, the pink molecules, to stay still at the right position. And then this thing gonna bring the phosphate group here and then push it towards your ADP, connect them together, right? So how this thing happen, how the phosphate, the pink color here can stay in the pocket because you have intermolecular interactions, okay? If you look at here, you have electron pair on the nitrogen atom, which is now ready to make hydrogen bonding with this carboxylic acid side chain of the amino acid in your ATP synthase enzyme. And again, you have NH2. And this NH2 is actually here in this direction, linked together with hydrogen bonding with the carbonyl group of your carboxylic acid. And moreover, I think H here can link with the electron pair of nitrogen here as well. And H here can link with the electron pair of the nitrogen in this adenine as well, adenine unit. So arginine, and this is the glutamic acid. Arginine and glutamic acid fix the structure of your ADP to stay still here in this position. And also you have aspartic acid over here. As you can see, we have OH with hydrogen bonding. They can stay right here. Okay. Now that is the binding site. So all over the place here is a binding site. So binding site is bind your substrate to stay here in the place. And then this is your active site. Okay. So active site will have the, as you can see from here, you have OH hydrogen bonding, OH hydrogen bonding around here, and magnesium ions as well can complex with two oxygen. As I said, there are so many proteins that have kind of metal ion inside, right? And the duty of this magnesium ion is to bring together these two oxygen from two different molecules and then collide them together. So this is the active site. So as you can see, everything here happens using intermolecular interactions, especially for hydrogen bonding. And also, this is ionic interaction between metal ion and the oxygen oxide ion, right? So this is how the molecules like proteins, very huge molecules, can control the chemical reactions. Okay, so if we, as you know, chemical reactions, if there is no catalyst, if there is no enzyme, they just randomly collide themselves together, right? So from kinetics of reaction, you know that with the right direction and enough energy, then two molecules can produce the product, right? But now the enzyme can help facilitate the reactions by bringing together two substrates to come together in the right direction, right angle, and collide them together, make the chemical reaction go forward easier. Okay, so this is the beauty of proteins. The blue colored things is proteins here, and I also show you the nucleotides derivatives over here. Okay, so these are relationships between some of biomolecules together. Now we have only one type of biomolecules left, the lipids. Okay, so let's talk about the lipid in the next flipped classroom. Thank you very much for this script. See you again later.